everybody, can we have a very warm welcome for Dr. Nim Tottenham?
Hi, good evening, everyone. I'd like to thank the Secret Science Club and the Dana Foundation for inviting me here tonight, and thank all of you for facing this freezing cold weather and making it out here tonight. I'm really glad to be here and share with you some of our research findings. The question that drives a lot of the research in our lab is an old question in psychology, which is how do we explain this link between experiences that we have early in life, both positive and negative experiences, and our emotional functioning later on in adulthood. And as you can all appreciate, this link is a really long one in humans. It can be enduring across a lifetime. And to understand some of the glue that might hold that link together, our approach is to study brain development. In particular, for this talk, I'm going to be focusing on the development of the amygdala and its connections with the prefrontal cortex. So although a lot of you may be familiar with the amygdala's function, if you're not, the amygdala is this deep set of nuclei in the brain that are responsible for helping us pay attention to when there's something really emotionally important in the environment and help us learn about that event. When we're feeling emotionally aroused, we tend to see higher amygdala reactivity. In adulthood, the amygdala has strong bidirectional connections with the medial prefrontal cortex. So the medial prefrontal cortex is this large swath of brain tissue that we all have right behind our foreheads. And it's really well positioned to gather information from lots of other regions of the brain, including the amygdala, and synthesize that information that serves in large part to help quiet or regulate the over-arousal of the amygdala. So those connections between the amygdala and the prefrontal cortex are, you can think of it like a bridge, a physical bridge connecting those two regions and communicating, sending information back and forth between the two of them. So why are we focusing on these two regions? Emotional behaviors, encompass the entire brain. But I'm going to focus on these two regions for the purpose of this talk because by taking this narrow view, we can discuss some of the principles that have been driving a lot of the questions in our laboratory. And we study the system as it's growing up. So another approach might be to just look at adults and look at the finished product of development. But instead, our approach is to look at the system as it's unfolding during development. So, like, I'm the type of person who, as the pie is cooking, I like to keep looking in the oven to see how it's changing. And sometimes you get different answers when you're looking at how a system is constructing itself over time. So one of the things that really motivates us is thinking about these early experiences. And why do we care about early experiences so much? There's a lot of reasons why. One of them has to do with the presence of sensitive periods during early life. So sensitive periods are these moments when our brain is first developing, when a certain brain system is particularly amenable to be influenced by the environment. So in other words, a brain region may take on certain biological properties that make it highly plastic and ready to be changed, shaped, sculpted by the experiences that the individual has. And if you use a metaphor, this is a window of opportunity or vulnerability of a system. The window at some point closes as we mature. And the phenotypic expression, or the way that we behave or our brain functions, should in some ways reflect the environmental influences that occurred during that sensitive period. So that's just one of the reasons why we have a particular interest in early life. So the emotional behaviors that we pay a lot of attention to in our lab are collectively called emotion regulation behaviors. And emotion regulation is this incredible skill that humans have. It's our ability to, first of all, have a strong emotion, but then be able to manage it or cope with that strong emotion. And as I'm going to be telling you, it takes a long time for that to develop. And parents of young children know that for sure, right? It's very hard to regulate our emotions when we're little, and hopefully we become more skilled at it across the first 2 decades of life. And when we look at a number of mental illnesses associated with poor emotion regulation, I'm listing a bunch here, what many studies find is that those illnesses tend to have their peak age of emergence or onset around the transition between childhood and adolescence. So that's the time when the diagnoses seem to really be coming online. So if that's the case, and we're interested in these early sensitive periods or moments when the brain is taking in information from the environment, then our approach is to look at the period antecedent to that emergence. So we want to really focus in on childhood and understand what is being taken in by the system during childhood that may give rise to these individual differences in emotional behaviors that we see during adolescence. So there are a number of ways we can approach this question, and I'm going to start by sharing with you a really simple study that we did with participants as young as 4 years old. My daughter was our first 4-year-old participant to make sure that we could do it. So from 4 years old to 22 years old. And we simply presented to participants pictures of fearful faces. And it turns out that when we look at other people's scared faces, it's a really good way to activate the amygdala because we're primates and we care about when other primates are feeling scared. So we present these faces to our participants. And I don't know if any of you have ever been in an MRI, but if you have, you may be wondering, how do we do this with children? And so I brought a video here. This is my other daughter, and she's in a pretend MRI. So we have a lot of practice in an MRI with young children, and we have to entertain them a lot. And so she's laughing because she's watching SpongeBob SquarePants right now. So she's got goggles over her eyes, and she can watch whatever she wants to. And then at some point, we'll shut off the movie and we'll present these fear faces to her. And we can, okay. We tell her we're going to do that. That would be a different study. So we tell her we're going to shut off the movie and SpongeBob, and we're going to put on these fear faces, and we can take pictures of her brain as it's responding to these fear faces. Okay, so that's enough of that noise. So we take pictures of everyone's brains, and then from 4 to 22, and then we smush them together in some statistical package, and we search the whole brain for regions that are changing in their responsivity to these fear faces as a function of age. And one of the regions that comes out for us is the amygdala. In this case, it's the right amygdala. So what I'm showing you here, oh, sorry, my arrow's pointing to the wrong place. What I'm showing you here is a negative correlation between amygdala activity, which is measured in something called a bold signal, and age. So to better view that, I'm pulling out the data and plotting them here with age on the x-axis. I'm sorry, I'll try and move around so you guys can see too. Age is on the x-axis from 4 to 22, and then this amygdala bold signal or reactivity is on this y-axis. And each dot is a research participant's amygdala activity. And what you can see is the younger participants, the younger you are, the stronger the amygdala signal is to these types of emotional stimuli. And it's not just these fear faces. Other groups have shown in response to other emotional stimuli that the amygdala at young ages seems to be really reactive. And at first that might be counterintuitive because we think of brain maturation as meaning an increase in function over time. But when we think about the amygdala's function, this pattern makes a lot of sense because one of the amygdala's main roles is learning about the relative safety and danger of the environment. And when you're little, and relatively new to this planet, you have a lot of learning to do about the safety and danger of the environment. So you want the amygdala chugging at a very early age. But then we can ask, using these same data, about those connections, that bridge with the prefrontal cortex. And I just want to take a minute to explain to you the technique of how one might do this. So we use a metric called functional connectivity. And so with functional connectivity, what you can do is you extract the activity or the time course from one region, like the amygdala, and you plot it over time. So sometimes it's going up, sometimes it's going down. And you can pull out the activity from another region, like the prefrontal cortex, and plot its activity over time. And the degree to which these two time courses are correlated with each other gives us an index of functional connectivity. So the inference is that these two regions are somehow in conversation with each other. And in adults, in healthy, well-regulated adults, what people typically see in response to things like fear faces is that the amygdala and prefrontal cortex are talking to each other, but they tend to be negatively correlated with each other. That is to say, when one region, like the prefrontal cortex, increases its activity, the other region, like the amygdala, decreases its activity. And that's indicative of an inhibitory relationship between the two. So to illustrate how that works, I'm going to walk you through this plot. So these are data from adults and older teens. And on the x-axis is that functional connectivity measure between amygdala and prefrontal cortex. So the more negative that relationship, the more inhibitory that relationship, the more the amygdala quiets down over the course of the scanning session. So this individual way up here had a very negative relationship or a very inhibitory relationship between amygdala and prefrontal cortex, and their amygdala quieted down the most over the course of the session. Those individuals also tend to have lower autonomic arousal. They tend to have lower trait anxiety, and so on. So the punchline is, for healthy, emotionally regulated adults, we tend to see this negative correlation between amygdala and prefrontal cortex. So now we can ask the question about its development. So with our same participants from 4 to 22, we perform this type of analysis. And here, what we see is, I'm showing you here not activity, I'm showing you this blue dot represents a metric of that connectivity changing with age. So to unpack that, pay attention to the bottom graph. So in young adults, 18 to 22, we see what I just described to you, that they're showing this negative connectivity value between amygdala and prefrontal cortex. That's what we would expect in healthy adults. In older and younger teens, we still see that negative connectivity, but you can appreciate that its magnitude is greatly reduced compared to adults, which is consistent with what we know about the slow development of emotion regulation. But what we became most interested in is what we observed in children, which was an absence of that negative connectivity. And if anything, we saw positive connectivity between amygdala and prefrontal cortex. So a quicker way to say that is, these data were suggesting to us that in childhood, the conversation between amygdala and prefrontal cortex was different than it was at points thereafter. Moreover, that switch, that developmental switch from positive connectivity to negative connectivity was correlated with behavior changes that we saw across age. So one of the behaviors that changes dramatically across childhood and adolescence is normal separation anxiety. So what's separation anxiety? It's when little kids don't want to be separated from their parents, right? They follow them around the house, the parent goes to the bathroom, the kid goes to the bathroom with them, and so on. And now I'm a mother of 2 teenagers. They are far on the other side of the apartment now. I never see them except when dinner's on the table. So we increasingly separate from our parents as we get older. And it turns out that this switch here statistically explains that behavior. So what I'm plotting here on the y-axis is separation anxiety. This is not clinical anxiety. This is just normal separation anxiety that all kids go through. And for those kids that showed that more childlike, non-regulatory, positive connectivity between amygdala and prefrontal cortex, they had higher separation anxiety than those children who started to show that more mature regulatory pattern. So in other words, there's a parallel between the maturation of this system, this emotion regulation system, and our ability to increasingly not need to be around our parents as much. So that's a prelude to what I'm going to be talking about next, the intimate role, the intimate relationship between parents and our brain development. Okay, so to take what I've been showing you and put it into cartoon format, in response to things like fear faces, in adults, what's typically seen and what we've seen is that adults' prefrontal cortex will have some kind of inhibitory relationship with with the amygdala. But in children, we don't see that. We see that the two regions are moving together. It's not a regulatory relationship. And so putting these data together, what we're seeing is if this is age in years along the x-axis, at least during childhood, we're seeing this very robust amygdala response to emotional stimuli. But it's happening in the absence of these adult-like connections with the prefrontal cortex. So that's made this period of childhood very interesting to us to think about moments when we might start looking for, narrowing our field of view for a sensitive period for the system. Moreover, we have some hypotheses that this very strong amygdala reactivity during childhood may actually be the biological prerequisite for the instantiation of these connections that are going to help regulate the amygdala someday. So if that makes sense, the idea is that this initial dysregulated amygdala actually kickstarts the process that grows the system that's going to help ultimately regulate the amygdala. And part of the reason why we think that is inspired by our work with children that have experienced severe stress early in life. And we've worked with several populations, but the data that I'm showing you here are from children who experienced early institutional caregiving. So briefly, we commonly call these places orphanages. But this is an institutional care setting that is a very stressful caregiving circumstance for an infant because, for many reasons, in part because the caregiving is done by staff members, not parents, who often aren't trained, and they're rotating shifts. They go home on the weekends or they get fired, and those caregivers are responsible for many, many infants at the same time. So it's an extreme case of neglect, of caregiving neglect for a human infant, and possibly one of the most potent stressors that a human infant can experience. And what we've seen in children who've had that type of experience is we see very strong amygdala reactivity to those fear faces. And that's consistent with a very long literature, a very rich literature linking early stress to amygdala hyperactivity. So that makes sense because the amygdala's job is to pay attention to things that could be dangerous in the environment. But that strong amygdala reactivity is coupled with changes in amygdala's connections to prefrontal cortex. And those changes tend to look like this in our hands. So these are the data I showed you before, where in our comparison group, the children with a typical caregiving background, children show this more immature brain profile that switches to a more mature profile in adolescence. But following early institutional care, children in the 4 to 9 age group show a pattern that looks more like the older adolescents. And so there's some hint that this strong amygdala reactivity may in part be accelerating the development of some of these connections to look a little bit more adult-like at an earlier age. So what we think is going on in typical caregiving circumstances is the brain develops hierarchically. So things that are low and in the back develop earlier, and then the brain kind of curls around like ram's horns. And so things that are in the front, like the prefrontal cortex, are going to take a long time to develop. And you guys know about the age that they think the prefrontal cortex reaches adult levels? 25 or so. That's the age, right? Which, the average age, right? Which car rental companies knew before neuroscientists, right? So this is going to take a long time to develop it, but it's hierarchical. So what we think we're seeing with our data, and this is just a working model, is that given that hierarchical organization, the amygdala in childhood may be driving a lot of the conversation to the prefrontal cortex. So I think about the amygdala as like the older sister that got there first, and she's instructing the prefrontal cortex about what she's learned about the environment. So that over development, over time, the prefrontal cortex can start to grow its reciprocal connections back to the amygdala. And that's what we see in adulthood. So based on fMRI data, we can't conclude that because fMRI is a correlational measure. But there are many rodent studies showing, using precise tracing methods and optogenetic methods, showing that that indeed is what happens, that the amygdala sprouts to the prefrontal cortex emerge developmentally earlier than the reciprocal connections, suggesting there's merit to this idea. So if that's true, then this reminds us of a 200-year-old quote by Wordsworth, which is, "The child is the father of the man." And so the reason I put this up here is because if we simply had an adult vantage point, we often think about the cortex as controlling the rest of our brain, controlling the amygdala. But from a developmental vantage point, we may appreciate that the amygdala early on is actually tutoring the prefrontal cortex about the nature of the world. So to start to approach this question in our data, like is what's happening earlier on influencing the way that the brain is self-constructing over time? So we use the data that I've been talking about so far, that fear-elicited connectivity between amygdala and prefrontal cortex, or what I'm calling here stimulus-elicited connectivity, in all of our participants. And we collected another type of connectivity, something called resting state connectivity. And resting state connectivity is the same type of connectivity I just described to you, but you collect it while people are doing nothing. And you would think while people are doing nothing, they're doing all sorts of things while they're lying in there. Like some people are thinking about what they're going to have for lunch, some people are thinking about how long until I get out of here, and so on. But despite all of those individual thoughts, it turns out that this network emerges called a resting state network, and there's one between the amygdala and prefrontal cortex as well. And the way that people think about this is that it's sort of like a functional skeleton of the brain, what the brain is doing when it's not asked to do anything in particular at all. And so that functional skeleton in resting state has been most closely linked with individual differences in emotion regulation that we see in adulthood. So we care about that resting state measure. Okay, so we collected both of those in all of our participants from 4 to 22, and then everyone went home for 2 years and came back, and we collected both of those measures again. And what we wanted to know is, what is the best predictor of future resting state connectivity? And what we found was the best predictor was not the way your resting state connectivity worked 2 years ago, but instead the way that your amygdala and prefrontal cortex co-activated in response to environmental stimulation. That was the best predictor of the way that the brain settled into its resting state connectivity. So we are thinking about this as sort of Hebbian learning, if you know about that, the idea that cells that fire together wire together. So it may be the firing of the amygdala and the prefrontal cortex is kind of a recapitulation of that idea. The way that they're co-activated is leading to the way that they're going to settle into the resting state. And in fact, when we pulled out that prediction over time and plotted it across age, what we see is it's the youngest participants who have the strongest prediction arrow from time 1, stimulus elicited, to time 2, resting state connectivity. And that is consistent with the notion that earlier on our brains are more plastic, more ready to change in response to the environment, but then they settle into a more stable pattern as we get older. So these data have really motivated us to more formally ask the question, so is childhood a sensitive period for the development of the connections between medial prefrontal cortex and the amygdala? And we've been inspired a lot by rodent studies, which have shown that stimuli that are learned early in life can be used later on in adulthood, often to reduce anxiety. When we're talking about humans, the parent doesn't have to be the biological mother. It can be the father. It can be an adoptive parent, grandparent. This is a learned attachment that we form. And so the caregiver comes in all shapes and sizes in the human. This is true for a lot of animals, that they spend time with their parents. It's often the mother in most species. But one thing that is obvious from this set of pictures here is that in the human, unlike most other species, we spend an extraordinary amount of time with our parents. So in most species, this juvenile period is on the order of weeks or maybe months. When you get a little kitten and you think it's going to stay like that for so long, and then you blink and it's a full-size cat. Well, we're not like that. We spend time with our parents on the order of years or decades, whatever the case may be for your family. And that's a very curious design for a species, because I don't know how many of you are parents, but it's a huge energy suck to raise another human being. And you'd have to ask, why would Mother Nature design a species like that? And it also is it's potentially not great for the offspring. They're vulnerable without that parent, and they have to hope that they're landing with someone good. So the argument that we've heard that we find most compelling about why such a design, we would have evolved to have such a design, is that this extended period of time with parents affords us the luxury of childhood. It allows us to stay in this immature state, and thereby allows our brains to stay in this immature state for a much longer time, which means we have a lot more time when our brains are highly plastic. So we can learn a lot more and learn much more exquisitely about our unique environments. So in other words, parenthood gives us this luxury of developing a highly complicated brain. And so parents provide, in many ways, a scaffolding for our central nervous system that allows us to have the protection of someone who knows something about the world while we're still learning about it. So again, we turn to the rodent literature to get some ideas about hypotheses we might make about the human. So again, I'm going to walk you through a series of findings from the rodent. This time it's a rat. And these are data from Regina Sullivan's lab at NYU. A young rat will learn to prefer its mother's odor, because they're blind and deaf still, so they rely on odor. And if you place the mother's odor at one arm of this Y maze, the young rat will, more often than not, head towards that door. They'll head towards that arm. They can also learn to prefer other odors if it was learned in the mother's presence. So in this study, Regina Sullivan's lab took a peppermint odor, which means nothing to the rat pup. It's a neutral odor. And they would pair it with something nice, like a brush stroke. And if the rat pup learns that in the presence of its mother, it will, and you place that peppermint odor at one arm of a Y maze, the rat pup will, again, prefer that odor because it's been associated with the mother and it's rewarding. That's perhaps not surprising. But the finding that is surprising is if that peppermint odor instead were paired with something not nice, like a mild foot shock, but the learning took place in the presence of the mother, this rat pup will still approach that peppermint odor. And so what this means is the developing brain is designed in such a way to learn a preference for things associated with its parent, regardless of the valence of that stimulus, whether it's punishing or rewarding. And that makes a lot of sense if you think about it. When we think about children's experiences with their parents, whether they're good or bad, children still form an attachment to the parent. It doesn't mean liking. It's not about liking. It's about the attachment process. It's about that glue that keeps us following our parents. So why is this happening? So it has to do with the amygdala. This is rat pup age on the x-axis. And there's a period in the rat pup's life, early in the rat pup's life, when the functioning of the amygdala is critically dependent on whether its mother is present or not. So if the mother is, and I'm saying mother because it's a rat, so only the moms are providing care. If the mom is in the nest with the rat pup, she decreases stress hormone production. In humans, we call it cortisol. In rats, it's corticosterone. She decreases cort production, which keeps the amygdala quiet during learning. If the mother is out of the nest, then all of those processes reverse and amygdala is active and it can engage in emotional learning. So when the rat pup learns that peppermint odor paired with the shock, if its mother is not there, so amygdala is free to participate in learning, this animal will avoid that peppermint odor, just like you and I would. But if it learns this association between the peppermint odor and the shock, and its mother is present, she keeps amygdala quiet, which allows competitive learning systems to take over learning. And if those systems are taking over learning, they're going to produce a preference behavior in the animal. And that's when you see the animal now preferring that peppermint odor, even though it had been associated with something aversive. So why would this be happening? The argument is that for the young animal who is altricial, meaning they depend on a caregiver for their survival, fighting and fleeing are not survival strategies that they can rely on because they can't do either well. Instead, the rat pups and our survival strategy at young ages is attachment, is to attach to somebody that is hopefully going to take care of us. And importantly, the parent has to be calm for all of this to work. So this mom is very calm. She is actually anesthetized. If she were displaying any kind of fear or distress herself, this would never work. Actually, she would be very, very good at doing the opposite, namely at amplifying amygdala reactivity in the rat pup. Okay, so attachment is the survival strategy of young altricial animals. And so I have a bunch of examples here, and I include a picture of Konrad Lorenz, if you remember this guy who raised the geese, to show that the attachment process is really learned, because the geese, there was no way they innately were going to attach to a human being, but because he was there and available, they learned to attach to him. I also include a video of my daughter We're here in an airport, and she's just following my husband through the airport. And I like watching kids at the airport because they just follow their parents most of the time. They just follow their parents when there's so many other things that they could be paying attention to. And it's that following behavior that we're really talking about here with attachment. It's kind of magical that it happens because it makes the attachment process so much easier. Okay, so we're biologically predisposed to learn to attach. We attach regardless of valence, or at least rat pups attach regardless of valence. And this attachment ends up becoming so important because it forms the foundation of so much of our subsequent cognitive and emotional development. So I've been talking about rat pups, and we wanted to know, several groups wanted to know, humans have such a stretched out developmental timeline compared to rats. Is it possible that we're seeing some of these processes stretched out over a similar time period? So I just want to use this picture here to illustrate what I'm talking about. If we think about species that are not altricial, they're precocial, like sea turtles, they don't need their parents. They're born prefab, right? They hatch out of their eggs, they look around for the moonlight, and then they paddle out to sea. They're small, but overall they've got the equipment that they're going to need. So they don't really have a lot of developmental plasticity. They seem to attain a lot of their adult characteristics at an early age. Other mammals, non-humans, who are altricial, they have this green period of plasticity early in life. So when we're thinking about the amygdala and prefrontal cortex, there is some moments of learning that are squeezed into that developmental period before they transition into the adult stage. And it's during that green period of plasticity when the caregiver is doing a lot of work to scaffold how the system is building itself. But in humans, we have a very stretched out green period, which gives us more time for the amygdala and then its connections with prefrontal cortex to cook, and more time for the caregiver to provide scaffolding. So would we expect to see some of these parental buffering effects of stress systems and fear systems at older ages? So these data here are from Megan Gunner, who's at the University of Minnesota, and she brought in children and she wanted to stress them out. So she gave them the task of delivering a public speech, which is a very effective way for increasing your cortisol responses. And children prepared these speeches and she measured their cortisol. And she did this either having them sit by themselves and prepare or sit next to their parents and prepare. And when children were sitting by themselves or with the experimenter, they showed this very nice mounting of a cortisol response, just like you and I would. But when they were sitting and preparing next to their parent, the parent actually buffered their cortisol response. Older adolescents also did this, and adolescents did not show this buffering effect. They showed a beautiful court response regardless of the person they were sitting next to, suggesting that this is some kind of, developmentally limited process. So we too were interested in whether parents were having a kind of buffering effect on these systems, but we wanted to go to that learning paradigm that Regina Sullivan's group had used in the rat pups. So we collaborated with Regina Sullivan's group, and with young children, we did this conditioning study. So we presented to 3, 4, and 5-year-old children a blue square, and it co-terminated with not a shock, but instead an aversive noise. So this noise was like loud, nails scraping down a chalkboard. So very aversive, and they think it's aversive too. And whenever they see this blue square, they would hear that noise. And the purple triangle was paired with nothing. So they learn these pairings either alone or with their parents sitting next to them. And then we put them into a human Y maze, and we asked the same questions about which direction the children would head. Would they head towards that awful blue square if they learned it next to their parent or not? So now I'm going to break this down because there's a lot going on in this study. Okay, so children learned those associations either alone or with their parents. They had to press a button. There was a reaction time measure involved in that as well. That gave us an index of how well they were learning the associations. And then, after the learning, the parents, if the parents were there, left. We put children into this Y maze, and then we gave them one more task at the end called a dot probe, which let us ask, were parents changing children's allocation of attention to one of those shapes? So now I'll walk you through just this learning part to show you what happens during the initial learning. There's also a lot going on in this figure. Okay, so I'll walk you through it. So ignore the difference between blue and gray for a second. So by all four measures, the reaction time to pressing and their accuracy, we measured their heart rate, we measured their skin conductance to give us a measure of autonomic arousal. By all of those measures, children were able to distinguish that CS plus, which was the blue square paired with the awful noise, versus the CS minus, which was the triangle safe. So children were able to learn those distinctions. Now, if you pay attention to the difference between blue and gray, gray is when children learned alone, and blue is when children learned with parents. And there was no effect of the parents during this initial acquisition. So children's behavior and their physiology looked the same whether their parent was there or not. Okay, so now parents go away. We put children into this Y maze. And, the Y maze, by the way, was like a play tent. And we put the blue square picture on one arm of a door and the purple triangle on the other arm of a door. And we told children to enter the tent and pick a door to retrieve a prize. And we showed them it was the same bucket of prizes behind both doors. We just wanted them to pick a door and do that 5 times. So when children conditioned alone without their parents, they more or less did what you and I would do and what the rat pups did, which is to avoid that blue square, which was paired with that terrible noise. But when children conditioned sitting next to their parents, they were showing this difference in their behavior where they were significantly, although modestly, but significantly approaching that blue square paired with the terrible noise. It wasn't paired with the terrible noise in the tent, but the blue square. So importantly, this was a within subjects design, which means these are the same children whose behavior is switching as a function of the condition that they learned it in. So remember that parents weren't having an effect during initial acquisition, meaning that parents weren't simply calming. We think parents weren't simply calming their children, but parents were actually changing the nature of what was learned and later recalled in this Y maze. And that effect of the parent was attenuated for children with higher cortisol levels, suggesting there's some biological constraints on this learning. Okay, the last part of this study was that dot probe attention task. So the way the dot probe works is children's task is simply to press a button to indicate which side of the screen this asterisk showed up on. It's a very simple task. But before that asterisk, we can present those cues to children that they had learned about. And their reaction time to pressing to the asterisks gives us an indication of where they were allocating their attention on the previous trial. And what we found was when children conditioned alone, their attention was allocated away from the bad square, which is consistent with what people find when people are using a dot probe task and presenting people with negative pictures. We look away. But when children were conditioned with their parents during dot probe, now their attention was allocated to that awful blue square, suggesting that part of what parents are doing may be helping to guide the child's attention towards what's important in the environment. So in the same blow, parents can provide some sense of security and safety, And what that security and safety does early in life is it enables exploration of things that might be a little bit scary. And that's really the basis of what John Bowlby described in attachment theory. So the last slide I'll show is we had a chance to scan children and presented them with cues of their parents. So we used pictures of their parents because we're very visual, and children are too. So we showed them pictures of their parents and contrasted that with pictures of somebody else's parent. And what we observed was in children, we get now parents decreasing amygdala response. So if you recall to the beginning of my talk, I showed you that children have a very strong amygdala response. But those data were collected without the parent being present or without parental cues being present. So here, now with a parental cue present, we see that amygdala is decreasing, which looks much like the parental buffering that we would see in the rodent data. Adolescents didn't show that same effect, which isn't to say that parents aren't important during adolescence, because they absolutely are. But it may be that at the level of the amygdala, parents start to lose their efficacy in changing its activity as much. And when we return to that connectivity measure between amygdala and prefrontal cortex, we see that the presentation of the parents are strengthening that negative connectivity between amygdala and prefrontal cortex. Again, adolescents are not showing that effect because they showed that negative connectivity to both their own parents and strangers. So in other words, one possibility of what we're seeing here is that when children don't have access to their parents' cues, we see this childlike, non-regulatory connectivity pattern between amygdala and prefrontal cortex, but with presentation of parental cues, we momentarily get these more regulated connections between amygdala and prefrontal cortex. And so one idea that we're playing with is that these routine interactions with parents day in and day out, even though they're resulting in these momentary regulated connections, over time are what help construct these more adult-like connections that we see in adults. And the strength of this effect of the parent on the brain was moderated by attachment security. So the more secure children reported feeling in their relationship with their parents, the bigger the effect the parent had on their brain. So altogether, what we think is happening is that during childhood, parents are really good at when they're calm, parents are really good at buffering amygdala and related behaviors, but when they're not calm, they're really good at amplifying them. Or when children are exploring off on their own, they may experience an increase in amygdala reactivity. But this up and downness of the system may be an important process that over time helps tone the system or teach the system about the parameters of emotional experience and associated neurobiology. And so we have some ideas about the neurohormonal cascades that get switched on and off as a function of the parent's presence or not. And the idea is that by the time the sensitive period, the putative sensitive period closes, and we look in adolescents or look in adults and we see individual differences in emotion regulation behaviors, that those differences are reflecting a lot of the learning that happened during this early sensitive period. So with that, I'll just tell you what I told you. So human amygdala prefrontal cortex circuitry develops very slowly in humans, but we are arguing that there's value in that slow cooking. And the transition to adult-like phenotypes we see starting at the beginning of adolescence, which gives us a very long sensitive period for the regulatory connections between prefrontal cortex and amygdala. And therefore, the environment can play a large role in scaffolding how the system constructs itself. And so parents may, in some ways, be this privileged information source to this deep emotional neurocircuitry that's developing during childhood. So with that, I do want to acknowledge a lot of people in the lab, some of whom are here tonight because this definitely takes a village to do these types of studies, and thank the families and individuals that participate in the research, and thank you for your attention.
My question is regarding the application of this research towards trauma in early childhood, and if a parent's presence and the attachment is not secure because of a parent's abusive behaviors or neglect, does that then condition the child as an adult to seek those kinds of environments? And then what are the applications of that in terms of, educating the public about the importance of keeping children away from abusive parents?
It's very complicated. It's a good question. In terms of neural mechanisms, I can only, speculate based on what's been shown in the rodent data. So if that rodent paradigm of pairing the peppermint odor with the foot shock were repeatedly administered, it becomes a model of abuse. And those rat pups will grow up and be more likely to show depressive symptoms. And the way that they often measure that in rodents is they do something called a forced swim test. So you see how long the animal will continue to swim, and animals that are depressed tend to quit earlier. To get to the question that you're asking, if that peppermint odor were reintroduced to those depressed animals, it actually has this paradoxical effect where it kind of rescues them and it reinvigorates them. And so, I think that understanding the neurobiology and understanding these mechanisms, and we can't say what happens in humans who are way more complex than rats, but I think that understanding how attachment works is very important in helping us understand the development of people who have been exposed to various forms of maltreatment, and trying to understand where is the plasticity in the system, what are the other sources of strength in a person's environment. So we've focused on the parent only, and that's a very blunt instrument. And so we have studies going on right now that are looking at the effects of teachers, for example, or other important people in children's lives. But I think the other part of that is, for me, it becomes important when thinking about the very difficult decisions that family courts have to make when children are potentially going to be separated from their parents. Getting courts to recognize very deeply that these attachments are very strong, even if they're in the context of maltreatment, and weigh that as equally as the many other things that the courts have to.
We have a question right here.
Yeah, hi, thanks. That was a really great talk. There was one thing, I don't know if you said it and I missed it, or if it's an open question. When you talked about the results of the children who were in orphanages, if I understood correctly, what you said is that at an early point, they were already showing the kind of downregulation, or whatever you want to call it, of the amygdala by the medial prefrontal cortex, which in some rough interpretation you might say is maturity. But presumably they don't develop very well emotionally. So what's the interpretation of that?
Yeah, that's a great question. So it is counterintuitive when you say it just like that. If I had the slides, I would pull them up. But in brief, you're right that it does seem counterintuitive that, this neurobiology is supporting emotion regulation and it's growing up faster maybe in kids that have experienced stress, which, by the way, has been shown across species as well following early life stress. And what might be happening is that the system, if you are getting an acceleration of development, then what you might also be getting is a truncation of that plasticity period. So it's growing the system up too quickly, perhaps. So what that might mean, and now I'm just speculating, is that you are not getting the same amount of time for all of the neural elaborations to reach language areas and semantic representation areas and all the areas of our brain that people recruit when we're engaged in emotion regulation. So what you may end up with by adulthood is and again, everything I'm saying is on average. I want to be clear about that. There are huge individual differences. So, but on average, what one might end up with following early life stress is a more reactive, reflexive, rather than reflective emotion regulation system.
Hi, thank you for the great talk. So I have a question about, so you're focusing on the effect of the negative experience in the presence of parents. Have you thought about the flip side, where you experience, whether the children experience a positive experience with our parents, where there are any different?
That's a great question, and we have because. Well, we were interested in that question too, and so there are unpublished data yet, but what we're starting to see is that parents are also influencing reward learning in young children. And I think all of this makes a good bit of intuitive sense. There are things that I can think back to from my own childhood that were positive things as well as negative things that I still kind of am drawn towards, right? So a benign example is like cigar smoke. I know that it doesn't smell good, but my father smoked cigars and I'm drawn towards it, right? My mother's Korean, so kimchi. I know it doesn't smell good, but I'm drawn towards it, right? So it's like, it reminds me of like that, those Proust stories of just being drawn towards things from our past, even though it's not about liking all the time.
We have one over here.
Hi. So I guess I was just curious, have you done any work into looking at the sort of what's triggering the closure of that sensitive period, the possibility of that being the onset of puberty, and that perhaps this entire sort of learning scheme of risk aversion is sort of going towards, looking at situations under which it's safe to procreate and situations under which it's not safe to procreate?
Yeah, it's a great question. I don't have an answer for you. I just have my hypotheses. So I think that the onset of puberty is playing a big role in this, but the way that I think that it is, or part of the way that I think that it is that it makes alternatives to your parents seem more attractive, right? So, like, again, I was describing my kids. At one point in life, when they were little, I had humans stuck to my body all day long. And then they got a little bit older, and they were crawling on the floor and I was reading a newspaper. And now they're down the hallway in their room and someday they're gonna be out the door, right? So making other people somehow more attractive than your parents is also increasing the physical distance between you and your parent, which might be another cue to the neurobiology to start growing up, because once you are separate from your parents, you have to start navigating the world independently. And you need that neurobiology to do that well. And so I think that, thank God for the things that parents complain about in adolescence, because we should have that separation from our parents. Otherwise, we wouldn't be here as a species, right? Otherwise, why would you want to leave your home? Someone's paying your rent, they're feeding you, they tell you you're really good even when you're not. And so, you've got to make the outside world compelling.
Yeah, so thank you so much. I have to apologize, I'm an engineer, so time series is important to me. When you showed the early graphs of the interaction between the amygdala and the medial prefrontal cortex, you showed this very much out of phase reaction. So when there's mediation between the prefrontal cortex and the amygdala, you see this very much upside down view. Mm-hmm. Now, can you talk about phase in terms of the time series? I mean, again, engineer, but are we talking reasonable time steps between when one might be initiated? If, in terms of, you show the fear face into the MRI, yeah. Is there a gap?
Yeah.
Because I want to say in terms of causality, you do expect a certain gap.
It's a great question. So disclaimer that I showed a cartoon time series, but your question is well motivated. So there are approaches, statistical approaches that people have employed that try and take advantage of any temporal lag between time series to try and infer directionality of one thing driving another. Those have been received with mixed enthusiasm, in part because, unlike other time series that you might be familiar with, this time series has pretty poor temporal resolution. So the bold signal relies on blood, and it's on the order of seconds. So some have argued that we really shouldn't be leaning too heavily on those temporal lags. But others have been very enthusiastic about that approach for the reasons that you're articulating.
We have a question on this side.
Hi, thank you again for your talk. I'm wondering, through your work and your research, what recommendations would you make with regard to reforms to juvenile justice, in a broader sense, with neurolaw being a new interdisciplinary discussion?
So thank you for your question. I tend to work with the younger ages than are relevant for what you're asking. But there is a lot of work in this area from people like B.J. Casey, who's at Yale, and Adriana Galvin, and many others who have actually been speaking in court cases to make the argument that actually the brain is still immature in the second decade of life, and maybe adolescence is longer than we previously thought. And so once people start to think about the adolescent in that light, it might change the way that certain sentences are delivered. And I think that that's having a much larger impact now than it has in the past.
We have another question over here.
Hi, I have a question about the implications for therapy and healing, given your studies, and how sort of fixed and concrete this window of sensitivity or plasticity is. Mm-hmm. I think therapists have found that mentalization and affective reflection are ways to kind of break the cycle of avoidant and anxious attachment passing on from parent to child. And that seems to be an interaction between the PFC then and the amygdala that's being promoted as a kind of plasticity. So is there a sort of therapeutic way of sort of enhancing or promoting a kind of plasticity?
So it's a great question. I think that some of the things that we're learning have implications for brain plasticity that can boost the work that is already being done by clinicians. So I think that the idea behind therapy is that it is working on brain plasticity, right? If the brain can't change, then it's pointless going to a therapist. So I think that if we can better understand the mechanisms of plasticity as it happens in childhood, and maybe how can it be, boosted up again in adulthood, that would be a promising direction for the interface of neuroscience and clinical science. There are studies that are ongoing that are looking at more pharmacologic ways to boost plasticity in adulthood. So if this is a longer answer than you're really looking for, but I think it's interesting. So if you, some individuals are born with amblyopia, so their one eye has weaker muscles than another, so their eyes might point in different directions. And so the treatment for that is to patch the stronger eye to get the muscles working in the weaker eye. And if you do that in childhood, then you actually change the way that the brain, you get the visual cortex back on track. So that the visual system develops correctly. But that patching intervention doesn't work in adulthood. because you're past the sensitive period. Well, you can make this model up in rodents as well, and it turns out that there are some pharmacologic agents like Prozac that actually reopen up plasticity in adulthood so that this patching intervention works in adult rats. And so this, it's not necessarily new in clinical science because there are many studies, and I'm not a clinician, so you jump in when I'm really out of my wheelhouse, but there are many studies showing that the combination of pharmacologic agents with talk therapy seem to have good effects for people, and maybe one of the reasons that's happening is because the pharmacologic agents are increasing plasticity, which allows the talk therapy to do its work and stick better.
We have one more over here.
Hi, thank you for the great talk. I had a question regarding to the age of adults that you use in your studies. So most of them I saw in the data were younger adults. Have you seen any difference between younger adults and older adults? Because though the prefrontal cortex, to my understanding, I'm very illiterate when it comes to neuroscience, but when it comes to the prefrontal cortex, it develops later, like by 25, but it's also the first to deteriorate, right? So like, does that affect emotional regulation in older adults versus younger adults compared to childhood?
So it's a great question. You're bringing up two things. So one, you're absolutely correct that the adults that we're using are 18 to 22-year-olds. In other words, those were the available adults that we have on a university campus. And arguably, those aren't the ages that we should be targeting. And in fact, I mentioned Adriana Galvin, who's at UCLA, and she, for similar types of study designs, actually won't use 18 to 22-year-olds, she'll use 35-year-olds, or an average of 35. And a lot of that is inspired by the work, again, of B.J. Casey's lab, showing that the adult brain is really not there yet, on average, in 18 to 22-year-olds. So that's one point. And the second question you raised is about, in aging populations. And it turns out, on average, in the context of healthy aging, that emotion regulation improves. And it's unclear why that is. There are a lot of theories. Some of them are neurobiological. Some of them involve, aspects of ideas of, a view on life that you have that's different than when you were 22 years old or so on. So that's a really active area of investigation, is emotion regulation in aging populations. Thank you.
Did your study look at or control for the effects of the parents' responses to the stimuli? Like, was it just their physically being there, like the anesthetized rat? Or if they said, ooh, wasn't that an awful noise?
Yeah.
With that kind of, where the child might be looking at the parent to see how to react to the stimuli?
Thank you for that question, because I didn't take the time to explain it. So in our study, the children were delivered the noise through headphones, and the parents were filling out paperwork, so they weren't interacting with them. So we started off with the bluntest instrument possible, which is just the parents there. The next question is the one that you're asking, which is, what about the individual differences in parents' behavior? Because some parents do have, this behavior, and another parent has another behavior, and that's certainly going to turn the dial in a different way. We wanted to start off with this first question about presence. We have another ongoing study where we're not fear conditioning the children, we're fear conditioning the parents and videotaping that. And then we show the videotapes to children, and so they see their parents' reaction to those negative stimuli. And so our initial data look like what's happening is that children are learning very well from their parents when something is bad, better than when they're learning from somebody else's parent that something is bad.
All right, I think we're going to wrap it up. Thank you so much, Dr. Tottenham. Thanks for coming, everybody.